Today we are going to talk about, I would not say the series of the translation, but yes, of course, we are going to talk about the protein folding, right? So in the last lectures, we have completed the translation in that three series, in that three lectures. The first we have done the translation as a normal introduction only. Then we have completed the translation in prokaryotes in the second video lecture. And in the third, we have completed the translation in eukaryotes, right? Of course, you can see it has been uploaded. You can see that in the separate videos, translation in prokaryotes, including that all of the enzymes and the releasing or the initiation or the whatever the factors. All the information we have already covered. So you do not need to go for some of the content to the chat gpt or some any extra information. Everything we have covered. Today we are going to talk about that after the translation, right? After the translation, what happened? The protein folding. Why there's a protein folding? Because of course in that translation, so today's lecture topic is your protein folding. It is of course one of the important topic of your zoology optional paper, UPSC paper 2, right? Okay, so about the protein folding, if I'll talk, protein folding. So during after the translation stage, after the translation stage, okay, the a long chain of amino acids, that's a long chain of, suppose that if we talk like this, so this is your one amino acid, this is your second amino acid, this is your third amino acid, and so on so on. A long chain of amino acid is formed, right, through the peptide bond. But this is not the actual structure in which the protein perform its certain or specified function, right? This is simple the structure of the polypeptide. Like this is simple the structure of that amino acid, chain of amino acid, which has been formed as a nascent product after the translation process. If you want to relate like that, so even after the transcription process, when the mRNA has been formed from the DNA, remember? So after that DNA, after that mRNA has formed from that DNA strand, in the transcription process, there is a proper step that is called the post-transcriptional modification. Of course, there's a significance of that post-transcriptional modification. So similar thing is the, like we do the guanylation, capylation, and the splicing. These are the mainly three factors. So similar process occur in the translation, not like the similar, but the modification, right? This long structure here, not there will be not the splicing or something, because the amino acid which is formed, the chain of amino acid which is formed from the mRNA, it has been formed after the post-transcriptional modification of the mRNA, right? So whatever the codons are required as the exons, right? Introns and exons are the codons, are the segments in that mRNA. Exons are those segments which are actually responsible for the coding region, right? While the introns is such just splice it out. That is unrequired segment. That is not coding for this specific. So here we are not doing anything splicing, but this long chain of amino acids need to fold in a specific kind of structure. Okay. So today we are going to, and this folding of that amino acid, it is not random, right? It requires certain enzymes. Then it is required a kind of chaperones, that is HSP70, HSP40, heat shock protein. Even the folding of that amino acid chain also requires some kind of enzymes and proteins, right? There are mainly three methods to fold the protein, to fold this amino acid, long amino acid chain properly. So that we are going to talk about the protein folding. In this lecture, we are not going to talk about that alpha helix and the beta pleated sheet and secondary and the tertiary structure. That is a kind of structure of protein we'll cover in that one of the next video. Okay, here just we simply talk about that with what are the associated compounds this long chain of amino acid folds into its structured proper function form. Got it? Okay, fine. So protein folding. So protein folding proceeds via and initial clustering among side chains of hydrophobic residues, okay, which prefer to be aloof from the aqueous environment, of course, which is prevent from the aloof from the aqueous environment. Clustering due to the non-specific interaction among the hydrophobic residues that lead the formation of a compact arrangement, that lead the formation of the compact arrangement, that is molten globule stage. Hydrophobic residues, what are the hydrophobic? Of course, basically now we know that, which is avoiding the water interaction. So that should be not at the border or the corner, right? Residues of the protein gather inside of the collapsed form within the core, and this collapsed state favored the formation of the secondary structure and encouraged the tertiary interaction among the residues. Suppose that this is your long chain of amino acid, it just fold like into, suppose that alpha helix, if you want to talk about, it is fold like that is the secondary structure. No, secondary structure is your alpha helix and beta plated sheet. Of course, in one of the video when we talk about, we'll talk about it also, how the formation of the alpha helix is folded so that hydrophobic amino acid comes inside that, right, to avoid that water interaction. Okay, so that particular assisted things are called, the process is called the protein folding. Okay, assisted protein folding, most proteins fold spontaneously to their native. Cells have a variety of mechanism to ensure proteins are folded correctly. Okay, by a specialized protein, of course, enzymes are itself a kind of protein, right, or the proteinaceous in nature. Then molecular chaperones interact with the partially folded or improperly folded polypeptide, facilitating correct folding pathways. Okay, HSP, that is heat shock protein, for example, HSP 40, 60, 70, 90 and 100 are highly evolutionary conserved family of proteins. Okay, number suffixes indicate the molecular weight. Okay, chaperones provide the micro environment in which the folding can occur. Okay, so this is something you can see if you if this is easy to see, or if it, or even if it is not, let me just elaborate this thing from you. So this is your long chain of amino acid. Okay, this is your long chain of amino acid. Here the chaperones comes like, okay, and HSP with the help of the HSP का full form ये है आपकी HSP, heat shock protein. Okay, they just associated and look, partially folded protein and this long chain of amino acid folded partially, right, then transfer to the chaperone. Chaperone is a kind of structure, like if you want to understand like that, chaperone is like this. Here it is a cover like structure. In that protein unfolded or the partially folded protein goes, and inside that protein fold properly, and then it take out in the proper functional form of protein. So chaperone is one of the associated things which help in the protein folding. Okay, then look, this is a chaperone, this is a folded protein. Okay, chaperone is a chaperone says HSP70 family of the chaperones binds the short sequences of the कैसे करता है hydrophobic amino acid से that emerges while the new polypeptide is being synthesized, shielding them from the solvent. Protein chaperones aid protein folding in two basic ways. The first is assisted folding and second is binding of that unfolded proteins to prevent the aggregation. Got it? यहाँ पे chaperones क्या है? HSP70, just a minute. HSP70 acts as a chaperones in the folding process together with an HSP70 60 or HSP70 chaperone. Central cavity of the HSP60 chaperone provides a sheltered environment in which the polypeptide fold until all the hydrophobic regions are buried in its interior. Okay, just prevent the protein aggregation. Okay, of course this is a specific mechanism. Two general categories of chaperones are recognized in the cytosol and the mitochondrial matrix. HSP70 is assisted by the HSP40 and other proteins to perform its functions, and that required the ATP. That means this process required the ATP hydrolysis. Okay, what are the enzymes? ये तो हो गया सीधे सीधे chaperone से. Then what are some of the enzymes which are also helping or assisting the protein folding? So protein disulfide isomerase. When we're going to talk about the structure of the protein, then we will talk about in your primary structure, in your secondary structure, what are the major bonds, in your tertiary structure, what are the major bonds. That particular form, this associate, that is also structure of the protein is also associated with this only, right? Gradually when we talk about the structure, you can relate much better in that way. Okay, so protein disulfide isomerase, protein sulfhydryl oxidase catalyzes the oxidation of cysteine residues, right? These are some of the information to mention in your copy or the notes, to form the disulfide bond. Cysteine form a disulfide bond. Of course, we know the cysteine is one of the amino acid containing the sulfur group with cystenyl residues by catalyzing the disulfide chain. So disulfide bond is mainly involved in the secondary structure or the tertiary structure, right? So long chain of amino acid folding with the help of that, so many other that R chain on the particular amino acid is helping. So primary structure is just making the peptide bond. Then secondary structure when we forming, that means except the peptide bond, there's the involvement of that R group of the each amino acid, right? That is making the secondary structure when the involvement occurs, right? Hydrogen bond and so many things, sulfur bond. Then the rupture of SS bond and its reformation with a different cysteine. Protein disulfide isomerase facilitates the formation of the disulfide bond that stabilizes the protein's native conformation. Okay. Eukaryotic sulfhydryl oxidases are flavin dependent. FMN, remember, flavin dependent. Dietary riboflavin deficiency accompanied by the increased incidence of improper folding of that disulfide D-containing proteins. Okay. Next is your peptide prolyl cis trans isomerase. Simply what you can do in that protein folding, you can talk about the three things: chaperones and the enzymes. Enzymes majorly आपके two enzymes है. Isomerase, all the X pro protein bonds where X represents any residues are synthesized in the trans configuration. X pro bonds of mature protein 6% are cis, as a 6 conformation is common in the beta subunit, and we know that alpha or beta subunit. And interconversion of the cis and the trans isomerase of peptide bonds with the proline. प्रोलीन का स्ट्रक्चर रिंग स्ट्रक्चर में NH के साथ दोनों तरफ कार्बन ऐड होते हैं by cyclophilins. Generally prolines are avoided in the alpha helix, we know that. While the cyclophilins participate in the folding of protein expressed by the viral invaders. Okay, so cyclophilins are pursued as a target for the development of drugs, and cyclosporin and alisporovir for the treatment of HIV, hepatitis C, and this. So what happens if this all associated thing, the chaperones and this enzymes and everything, will not function, any of the stage will not function properly. Suppose that if this is a proper structure folded form of the protein, if, and this is actually the we talk about the functional domain also in next lecture. So that means in that quaternary or the tertiary structure in which form the protein is functioning, there's a proper domain that is called the functional domain, which is a functional area or the space of the protein. Agar if there will be a problem in the folding of the protein, then there are some of the diseases which can cause, है ना, and that misfolding or improper folding because of that less or improper functioning of chaperones or the enzymes. So this in the next series of this only, we are going to talk about some of the diseases of the protein misfolding, right? So in one of the diseases, and connect the concept of the prions and the related neuro diseases. Okay, neurodegenerative, the neuro problem diseases. Okay, so that is one of the questions recently came in the 2025 thalassemia, I think, one of the disease, and the sickle cell anemia we're going to talk about in the next lecture. Thank you everyone.